Ma'am, recording has been started, ma'am.
Okay, Sindhu, we'll start the program.
Okay, ma'am.
Yalini,
Ma'am?
we'll start. Welcome,
Yes, ma'am,
Mr.
I'll start.
Ashok.
Yes, ma'am.
Emma, resource person கேட்டுக்கலாமா அவங்களும் ரெடினா we'll start.
Yeah, I'm ready.
Okay. Ma'am, shall I, ma'am?
Yes, ma. We'll.
Good morning to one and all present here. I cordially welcome all of you for the guest lecture conducted by Department of Biotechnology, sponsored by DBT Star College Scheme. The topic is replication pattern in prokaryotics and eukaryotics. I cordially welcome our chief guest and faculty members and the students for the guest lecture. Now I call upon our HOD ma'am to give the welcome speech.
Good morning, all. I take a great pleasure in welcoming all for this guest lecture on replication process in prokaryotes and eukaryotes. I welcome our correspondent and principal sir who were behind our success in all our endeavors. And at this juncture, I would like to welcome our resource person, Mr. Ashok Dhinakaran, doctoral research candidate, Toronto, Canada. I'm really very happy that he accepted our invitation, and we are aware that definitely this is going to be an untime for him because of time zone difference. So thank you for accepting our invitation, sir, and giving a talk on the replication process in prokaryotes and eukaryotes in a very simplified way. And I also welcome our faculty members of Department of Biotechnology, our DBT Star College Scheme Coordinator, Dr. A.K. Vidya, and other coordinators of other departments, Star College departments. And I'm very happy to welcome our students who are really very energetic and enthusiastic to attend this program and gain knowledge in the topic. So, I welcome all. Thank you.
Thank you, ma'am. Now I call upon our faculty, Miss Sridevi ma'am, to introduce the resource person.
Sridevi?
Ma'am, good morning, ma'am.
Yes, ma. Good morning, ma.
Sorry, ma'am, I got some disconnection only. Sorry for the delay. Good morning, everyone. I extend a warm welcome to all, and I find immense pleasure in introducing our resource person, Mr. Ashok Dhinakaran, doctoral research candidate, Nanobio Interface Facility, Ryerson University, Toronto, Canada. And let's see his profile. He completed his undergraduate degree in biotechnology from Rna University and master degree in nanobiotechnology from Hindustan University. He is a recipient of Canada's Ryerson International Student Scholarship for a fully funded PhD program, and he is a doctoral research candidate at the Ligation Knowledge Institute of St. Michael Hospital and Ryerson University, Toronto, Canada. Let's see his research profile. He published 5 publications in international journals and has citations of 62. He is currently doing his research area in 2 parts, one in nanomaterial fabrication and application for cancer metastasis detection and therapy, and another one is COVID-19 detection and testing of antivirals. And good morning, sir.
Good morning, madam. Thank you for your introduction.
Okay, sir. I will hand over the session to our resource person. Sir, you can continue, sir.
Okay, thank you, Ms. Sridevi. That was a very elaborate introduction, and welcome you all, guys. Okay, let me turn on my camera. Okay, so yeah, I extend my welcome to the HOD of the department and my friend Sindhu and all the students, and also the person who has given me the introduction. And Saturday morning, all of you might have been in a weekend mode, and I'm sorry to disturb you with all my technical talk. But still, this is the only time that I could actually spare because it's a night time here. And without any further ado, let us directly go on to the talk, okay? So, okay. So just to get you guys into the mood, I have just made some few slides regarding what we are doing and where I am located here, and about my university. So it can be a little bit refreshing. It's going to be quick and a few minutes about it, if that is okay. And okay, so yeah. Ryerson University, it was established in 1948, and it's a public research university. It's located in Toronto's heart, which is the Yonge and Dundas Square. It is the Yonge Street it was the world's longest street, so the university has its credentials to be located in such a nice location. And yeah, and okay, how does our university look in different seasons? So this is a nice warm summer, but unlike in India, we get summer here only for a month or two. So we have to enjoy while we can enjoy. So this is the fall season where it is already beginning to shed all the leaves, and it's going to be really cold. So it marks the beginning of winter, which people don't like here really, but we have no other go. Toronto has been blessed with winter for more than 6 months in a year. So it's going to be fully covered with snow, and all we can see is the color white everywhere, as you can see in these pictures. And yeah, so we can't get enough of winters here. It's going to be really bad. If you don't love snow, you won't love Toronto. And summer is the best part, but it is only for a month or two. And the nightlife at Ryerson and Toronto, it's going to be really awesome. So as you can see, it is all well lit with colors and bright, buildings and lights. So it is like more of a cityscape. And where I actually do my life sciences part of research, that is the cancers and coronaviruses, it is all here in St. Michael's Hospital. It is a teaching hospital where students like us go and do research. So it is like an international and world-class facility which has all the current and up-to-date equipment which we can actually do, and it is now doing great in COVID research as well. And I'm really blessed to be a part of COVID research in finding an early detection tool, and also for aircrafts to find out antiviral coatings. And that's about me and where I come from and what I do. So let us quickly jump into the actual topic, which is going to be replication of DNA. So I just want, I don't know which year you guys are all in. I think you guys are all from the first or second year. And if you have been introduced to nucleic acids, DNAs, and their structure and all that. So just for a brush up, I've given this slide here. So as you can see, it is a nucleic acid molecule which contains a sugar and then a phosphate group and then a nitrogenous base. So there are two types of the bases, which are pyrimidines and purines. So the purines are guanine and adenine, and cytosine and thymine are your pyrimidines. So from moving forward from here, let us just name them as A, T, G, and C, so it's easier for us to talk instead of telling the full name every time. And okay, so one end of your nucleic acid is going to be a phosphate group, and the other end is going to be a alcohol functional group, which is OH. So these mark the two ends of your nucleic acid literally. Okay. So here, this is the elaborate structure of a DNA. So you have your double helix in here, and they are intertwined with the A, T, G, and C components. And as you can also see, you all always know that A pairs with T and your G pairs with C, and there is going to be a double hydrogen bond between A and T, and it's going to be a triple hydrogen bond between your guanine and cytosine. So when you see the end that contains your phosphate group is named as 5 dash, and the end that contains your hydroxyl group is named as 3 dash. So that is the convention how we call the double-stranded DNA. And you also know that DNA is of 2 strands, which are complementary to each other. Complementary is nothing but one is the exact mirror image of the other. So it is how it is going to be. So when there is going to be a gap, it is going to be filled by this. Okay, so and also this is going to be filled by this. So that is how complementary they are. So which how to say that is exactly like a lock and key model where one lock can fit only that key. So it is going to be like that. Okay, and okay, so this is the complete structure, and this is only your one part. I mean, so okay, so imagine it is going to be this part alone, which is magnified and shown here in this image. Okay. And students, at any point of time, if you have any questions, you can unmute yourselves and you can ask my question. Ask questions to me, and also let me know if I'm going too fast or if I'm going too slow, and so that accordingly I can adjust. Okay. And at any point of, I'm just in an assumption that you guys have already know the structure of DNA, and we can directly jump into the replication. But if you don't know anything at any point of time, you can always stop me and ask. Okay. Then, okay. So what actually happens in a DNA replication? So replication happens when a cell is being split into two. Okay, so you know cell division, there are going to be a lot of stages in cell division. So initially the nucleus is going to be split up, right? So when the nucleus splits itself, the DNA within the nucleus also splits itself. And how that happens is what we are going to look today. And it has a lot of steps. And here our topic exclusively talks about prokaryotes and eukaryotes. So prokaryotes are very simple, as you know, in their structure, because they contain only a few thousand base pairs, whereas your eukaryotes, say for example, your human DNA, it's going to have like millions and zillions of base pairs. So eukaryotic replication is really complicated, and scientists around the world are till date trying to find out the exact working mechanism of your eukaryotic DNA replication, but they haven't completely decoded the actual mechanism that is lying inside. So moving on to the prokaryotic. So, okay, so just let me tell you the difference between prokaryotic and eukaryotic. More than the differences, similarities are going to be more. So prokaryotic replication, if you know how it happens and what happens in it, it is what is exactly going to happen in a eukaryotic replication as well, but except for a few changes here and there. Okay, which I will be letting you know at the end of this presentation. So first, let us cover the prokaryotic DNA replication, because it was easier for the scientists to figure out the whole mechanism that was lying inside the replication process. So the first step would be initiation of your replication and unwinding. Next, it's going to be okay, this is actually not a step. And then you have your primer synthesis, and then elongation, and then finally termination. So this is the basic process that happens in most of the molecular processes inside a cell. Okay, so moving on to the first step, your initiation and unwinding. So for every step, there are a lot of enzymes that are involved. So without these enzymes, it is not going to happen. So these enzymes are the actual driving forces for your replication process. So the first step would be initiation. let us see. Here they have considered a plasmid DNA, which is circular. So prokaryotes, I mean, the common examples for your prokaryotic organisms would be your bacteria. So, you know, bacteria has a simple DNA, I mean, double-stranded, and it's going to be circular, right? So your circular DNA here, so imagine it's just going to be a double-stranded circle, and when it is time for the bacteria to divide itself into two, it is your binary fission. Before that, what happens is something like this happens within the plasmid. This formation of your replication bubble is called initiation. And how it begins, and when it begins, is that is told by the cell, I mean, other signaling receptors to the nucleus. I mean, sorry, your prokaryotes do not have a nucleus, but they have other signaling mechanisms which tell them that the cell is ready for replication. And at that point of time, the replication bubble is going to form, for which your first enzyme comes into picture, which is going to be your helicase. Okay. Okay. So as you know, helix or any helical structure is just going to be intertwined. So as you can see, the cable, like a wire in your home, when you have a few old appliances, when you see twisted pair of cable, so they will be like intertwined like this, right? So anything like this, you can call them a helix. So an enzyme, helicase, is responsible here for the formation of this bubble. So what actually the helicase does is goes and breaks down the bond between the two bridges here. Okay, so the hydrogen bonds are broken by helix. Sorry, the helicase. So what happens is the double helical structure is unwinded here. So your first step is going to be initiation and then unwinding. And you might have a question here and ask if it can happen at any point in the circle. No, it happens only at a specific location in the whole genomic DNA of the prokaryote, which is called your origin of replication. There is a specific location in every gene, every genome, which says where it has to replicate. And that is called origin of replication, and it is a gene. So in short, they have named it as origin. Okay, so it's more convenient for us to, you know, use that gene. And every genome of any organism, you can find origins. Okay, and for prokaryotes, it's going to be one origin, which is one origin of replication, whereas in eukaryotes, it's going to be multiple origins of replication, which means it begins and it happens at many points. Okay. So imagine there is going to be many replication bubbles within a DNA. But I mean, this image is not representative of a eukaryotic DNA because it's going to be linear, as you know. Okay, only prokaryotic DNA is circular. Okay, so as the replication process happens, what happens is your bubble grows at both the ends, and finally what happens is it completely comes off from this and forms two different DNAs. So it is just like peeling off a sticker, right? So you have a sticker, and then imagine it's just one, and at the beginning you will create a bubble, right? So in order to peel off skin, you have to create a bubble at one point of your the sticker, so that the rest becomes easier and very smooth, right? You know what I mean? So like that, at the end, you will have two, but exactly of same sized circles. So just imagine it to be like that. Okay. So that small portion which you give in the beginning is called your origin of replication and initiation and the replication bubble. Okay, if that helps. Then, so what happens? Okay, from the inside, we saw the helicase, the role of this enzyme, which is going to break down the, okay, let us concentrate only on one half of this, okay, because the other half is exactly the mirror image of one half, okay. So talking about two things simultaneously on both sides might be a little bit confusing. So it is better to just look at it in one direction, so it's easier, okay? So here, your DNA helicase breaks down the hydrogen bonds between your bases here, right? So bases would have been like this, now it is all broken. Next, there is another problem here which is involved at the other end of your replication fork. Okay, so this place is called a replication fork. Okay, so here on the other end, what happens is, imagine you have a twisted cable or like a twisted twine thread, and you start removing it into two. Okay, just with one person, if you try to remove it, you'll be able to do it only to a particular extent, and after that it is going to be intertwined and it's going to be tangled, right? So that tangling is a problem here with your helical structure. In order to remove the, in order to do it in a successful manner and remove the twine into two different threads, it is easier if there is another person on the opposite. I just wanted to make sure if you guys are able to see my video. Someone, can you unmute yourself and let me know if you guys are able to see me?
Ashok, video is not visible.
I'm sorry, what? Hello?
Ashok, I could not see the video.
Oh, you guys are not able to see me?
No, Ashok, I can see only the screen.
Hello. Oh, okay.
Sindhu, I am able to see the screen and also Mr. Ashok.
Okay. Okay. I guess
also.
if you are looking from a laptop or a computer,
Yes, ma'am.
then
Even I can see.
Yes.
Okay.
Okay. If people are able to see, it is good. Okay. Thank you. Because I was just making some actions with my hands, so I just wanted to make sure if people are able to see me. That's it. Okay. So on the other end, if there is another person standing, and then if they are helping you to coil the thread in the same direction in which you are unwinding, then you will be able to remove the two threads separately successfully without any tangles in between, right? So this red portion here that you see, let me go back to the bigger screen. Okay, this enzyme that you see here is one such enzyme. So it is a class of enzyme which is called topoisomerase, and it is a gyrase. Okay, so this enzyme, what happens is, as the helicase keeps splitting the two ends across the replication fork, what happens is this one keeps winding in the opposite directions, so that the stress that is accumulated in the corner is keep on be relieving. So one end they are cutting and the other end they are twisting, so that the two strands of your DNA come off easily without any tangles. Okay, so the two enzymes in your first step would be a topoisomerase and then a DNA helicase. A helicase breaks down the helix. The isomerase or the gyrase relieves the tension in the strings. Okay, I think that's a very easier step. Okay, so now what happens is, another enzyme or another protein comes into picture, which is called your single-strand binding proteins. Single-strand binding proteins. So what happens is, these proteins come and bind to single strands. They do not have the capacity, or they don't have the specificity to bind to double strand. They can only bind with a single strand. Okay, that is their specificity. So what happens, and what is the role of these single strand binding proteins is, once you have removed, once helicase has cut these two, there is going to be only a small gap between the two strands. Okay, and you always know that A has a good affinity towards T and your G has a very good affinity towards C. Okay, two best friends, no matter how you try to split them, if you leave them alone, they will again try to join, right? But there has to be another group of friends who keep them apart and who keep them from talking to each other always, right? So that group of friends who do not allow the separated friends to talk to each other, they are called your single-stranded binding proteins. Okay, so the role of these guys is not to allow them to unite back. Okay, so the three components in the first step would be your isomerase, helicase, and then your single strand binding proteins. So the names, if you know what the meaning of their names are, you can easily associate their roles as well. Okay, then let us move on to the next step. Okay, so this is the overall depiction of DNA replication, which we will come back in the quickly. I think that is the step that I have to come immediately. Okay, so let us then look into here. So the second step is going to be your primase. Okay, so primase is another enzyme here, as you can see, a DNA primase. So the first, once the replication fork is created, and okay, so let us be clear about which is the original strand. So this 3' and 5' is going to be the initial DNA that we have till here, right? And this is going to be your daughter DNAs. Okay, so forget about these two parts for now and focus only on the original DNA. Okay. Okay. Okay, so here what happens is immediately once they are split, so these violet colored things are your SSBs, okay, your single strand binding proteins, and your blue is your helicase, and this is your topoisomerase. Okay, so all that is clear, and immediately comes the next enzyme, which is DNA primase. So what is the role of DNA primase is to let this RNA primer to come and bind into the position. First, let us go back to see what a primer is. A prime, okay, so the meaning of the word prime is first, or like one, or begin, or start, or primitive is like old, or the first thing that came into picture. Your prime minister is the first citizen. So it is like that. So once you know the word prime, here what happens is that marks the first step of your DNA replication, where the primase is an enzyme which facilitates your primer binding. Okay, so a primer is something which helps to begin the reaction. Okay, so the whole DNA replication process is a chemical reaction. Okay, and the I don't know if you guys have come across the polymerase chain reaction in your chemistry, in your schools, or even in your college. So polymerase chain reaction is when you have identical single units which are combined together in a continuous fashion to form a bigger unit. It's exactly the same here. What you have is individual DNTPs, which is your nucleotides, which are your A, T, G, and C, which are going to be continuously arranged and formed a bigger nucleic acid component. Okay, so once your RNA primer is just a small or short sequence which has some 10 to 15 base pairs. Your base pairs are nothing but your complete A, T, G, C with the phosphate and with the hydroxyl group and everything. So the whole setup is your base pair. So this primer has 10 to 15 base pairs, which comes and binds initially to here. But it cannot just come and bind by itself. There you need a enzyme which facilitates, which is going to be your your DNA primase. Okay, and how the primer has to be. There are conditions how a primer has to be. So a primer has to be exactly complementary to your DNA sequence. For example, if your initial sequence is something like this, okay, then your primer, okay, so it extends in both the direction. Let us forget about it. So your primer in this case should be TAAA and then CCC. So this short sequence is called your RNA primer. And how will the system know that this should be the sequence? That is instruction is what is given by your DNA primase. So your primase directs the nucleotides in your nucleus to come and bind in its place in exactly complementary fashion so they can begin the reaction. Okay, so the first step is going to be your RNA primer. Once it comes and binds here, then the next enzyme comes into picture. So the next enzyme, okay, let me clear the screen off. So the next enzyme immediately after your primer is coming and attached with the fork, then the one that you need is your DNA polymerase. Okay, so polymerase is nothing but an enzyme which enhances polymer chain reaction. Okay, so it is going to enhance the process, or it's going to make the process happen. Okay, so what happens is in your nucleus you have your A, T, G and C all in abundant. It's going to be present more in number inside your nucleus, and then your DNA ligase is a person, or it's an your DNA polymerase is an enzyme. It just puts the puzzle into its place. So imagine you have given a lot of pieces of your puzzle which can go into exactly one place, or it can go only into, I mean, you have A, T, G and C. You can put A's only on T's and G's only on C's, and vice versa. In that case, the person who goes and puts everything into the places, it's going to be your DNA polymerase. Okay, so DNA polymerase, once your primer is there, your DNA polymerase knows what to do, and it continuously extends the cycle. And what happens is, one by one, they come and join and they form a continuous sequence. Okay, but now there has to be a lot of problems in this. As I've told you, it is not that simple. There is going to be tiny tweaks here and there which needs to be done, which are, the first one, it is going to be your okay, what direction does your DNA polymerase go? Okay, your DNA polymerase always goes or grows in the direction of 5' to 3'. This is the convention. It can grow only in this direction. It can never grow from 3' to 5. No. Okay, so as you can see in the picture, right? So this end is 5. Okay, and as you can see, the 5 grows here and it goes all the way, and this end will be your 3'. Okay, whereas here again, your the direction of extension is going to be from 5' and it goes all the way and it forms 3'. Okay, let me come back to these. Okay, one more thing that you have to note is the upper and the lower arms of your DNA, they do not replicate in the same fashion. There is going to be 2 different patterns for your upper arm and the lower arm, which we will come into the next. Okay, so one thing that you have to keep in mind is your DNA polymerase can run or can grow your DNA only in this direction, sorry, 5' to 3'. It can never go from 3 to 5. And the reason being, why does it go only from 5 to 3? Again, that has an explanation. Okay, so which is, you have to go back to your structure of DNA for that. Okay, okay, let us see here. This is going to be interesting and more elaborate, so you can actually easily understand. So what happens is, okay, the gray color arrow shown here is your template strand, which is the original or your mother DNA, whereas your red color is your, what to say, it is your new strand that's being formed, and it's growing in the direction of 5 to 3. Okay, so what happens is, when you go back to your structure of DNA here in the beginning when I showed you here, you know that the phosphate end is your 5' and your hydroxyl end is your 3'. What happens in every step is, your one phosphate can go and attach only to another hydroxyl end. So for this replication to begin, the primary requirement is a free hanging hydroxyl bond. So a hydroxyl bond has to be there protruding outside, from which you can actually link more and more stuff, right? So only when you have a hook in the first place, you can put links of your chain and you can keep building it. So first, if you do not have a hook, if it is a ring and you have another ring, you won't be able to form a chain at all, right? So in this case, your hook is going to be your OH group. So every OH is a hook. So at the end, there has to be a hook, so you can always put a ring in it, okay? So assume that your phosphate is going to be a ring and your OH is going to be a hook. So it's going to be that way, so you can always put them one after the other, right? If you put a hook and hook together like this, and two ends are going to be your rings. So you cannot extend the rings on both directions, and the reaction stops right there. So you alwa' have to put a hook, a ring, and then you continue the process like that, okay? So here, and that's the reason why it can grow from a 5' towards a 3', okay? Okay, so as you can see here, I will show you the actual reaction, how it happens. So you have your OH group here, and then you have a phosphate group here. So phosphate is going to be always ATP or GTP. So you have your adenine, guanine, cytosine, and thymine, and all these TPs are your triphosphates, which is shown here in this three spheres, okay? For this reaction to happen, or for this combination to happen, it needs some energy. And you always know that your phosphates are energy packets, right? So your 2 energy is cleaved off in this step. As you can see, 2 phosphates are cleaved off. So what happens is when you remove 2 phosphates, it gets energy. Using that energy, the OH and this phosphate combines. And once they combine, this is what happens. As you can see, phosphate is gone out and these ions are utilized as energy for the process. And in the meanwhile, there is a bond that is formed between the 2 sugars and the hydrogen bonds are eventually formed, right? So that is also there. Now, going back to this. Okay, going back to this. First, let me show you. Okay, now I have told you the actual molecular mechanism of how an A pairs with T and G pairs with C, and how it extends from the 5' to 3', and how the replication fork happens, and how the cycle extends. Now let us get into the problems. Okay, so anything has to have issues, right? Nothing can happen without issues, and there will be people who can create problems. So we are going to look at them here. Who is your anti-parallel strands? So your anti-parallelism. So you have a lot of people who always rebel, right? So when people run in one direction, the other group runs in the other direction, and these are the ones who create the issues, right? So let us see. And so far, are you guys following? Is it comprehensive, or do I have to go slow, or is it okay at this speed to continue? Students? Okay, so taking that as a response that you are able to follow me, I'll just go ahead and continue with the same pace. And at any point of time, again, if you guys feel that I'm going a little bit faster, or if I need to move fast, that you guys already know a few of these stuff, and if I'm going over it again and again, it's a kind of, repeated, just let me know, okay? Then unidirectional replication, the replication can happen in only one direction, but there is another concept here which you have to address, which is anti-parallelism. Now, what happens is the direction of winding replication is here, but what happens is one string runs from 5 to 3, and the other string runs from 3 to 5, okay? Now, let us see here. Your green shows the proper direction, which is from 5 to 3, and it's also in the direction of your unwinding and initiation. So that is easier. So what happens is, as and when this bond is broken here, what happens is your this DNA polymerase keeps on running in the same direction, and then it keeps adding your molecules. And as you can see, in every step, the green arrow keeps on growing along with the unwinding, right? So as and when it unwinds, the green portion continuously forms your DNA. So this is now your new first strand and second strand, and this whole is going to form a DNA at the end, and this half is going to form a DNA at the end. But as you can see here, the orange stripes, they are intermittent. They have gaps in between. And what's the reason for such gaps is they have to run in the opposite direction. They do the hard work here while the green arrow does the smart work, or like a easy work, or just running with the flow of the process in the direction, right? So what happens here is when you look into the actual process deeply, right? Let me go into this picture so I can show you. Okay, screen is clear. Now, as you can see here, the direction of motion is here, and this guy keeps on replicates and then keeps going, moving forward. So what happens is your DNA polymerase keeps on goes in this direction and adds, and this is done. Going to this portion, what happens is first the primer comes and binds here, right? And then what happens is they grow in a particular direction. Imagine that they started here, the DNA begins here, okay? And once it is cleaved, I mean, once it is broken, they form a RNA polymerase, and from here it runs and it completes the strand. Then, meanwhile, your helicase is not going to sit in one place. It is going to keep on be cleaving this DNA into double strand into single strand. Then what happens is this region is left out. So as a continuity, as you can see here, when this guy runs, he keeps on running as and when this opens up. But what happens is this guy starts here and he runs and he finishes. But what happens is he has left a gap in here, as you can see. Then what happens is another RNA polymerase comes and binds here, and then it goes and completes till here. And then there is going to be the DNA helicase is still going to divide this, and there is going to be another gap. So in order to fill this gap, another RNA polymerase comes and he tries to fill the gap. So what happens is this process is going to be intermittent because it has to, there is a rule that they can grow only in this direction, right? If this strand can grow in 3 to 5, then he can also run like the green and they can continuously form. But that's not the case here. That is because the initial DNA strands are anti-parallel and they are complementary to each other. They are mirror images and they are not exact superimposed images, right? So what happens is these are fragments that are formed. Initially, when they were studying DNA replication, people thought that it was continuous, but then there was a person from Japan. His name was scientist Okazaki, and he was the one who found out about these intermittent fragments of DNA. Then people named it after him, which are called your Okazaki fragments, as you can see here, right? So the fragments, okay, so it is going to form from here, and then as it grows, there's going to be another gap. And what happens finally is these patches are adjusted. I mean, all these small fragments that are formed will be finally run by one person who can actually finish or seal these gaps, right? So imagine that imagine the whole thing like your zipper flyer in a bag. So what happens is when you run the zipper, so that is going to be your helicase, right? So it opens up the two fragments of DNA into two halves, right? Your single stranded. But when they are zipped up together, they are going to be a single, they are going to be a double strand. But when the flyer runs across the zip, then it's going to be two single strands. And imagine in your jackets or in your raincoats or in your hoodies, you have these runners that you can attach at any point, right? I mean, you can cut open the the runner and the strand, and then you can put it back together. You assume once after you remove one, you're trying to put another runner in place. So one runner which runs in the direction can easily be closed. But imagine a faulty zip which has gaps in between. What you can do is you have to somehow try to move the flyer at one end. That is going to be a very difficult process. But once you have moved completely with all the gaps and you finally zip it together, it all comes back into place, right? So that is how it happens in your second half. So here, the names of these strands, to make it more understanding, they have named this strand to be your leading strand, as it is given here. And this strand is called your lagging strand. So your lagging strand takes some time to finish the process, whereas your leading strand completes it as and when your helicase cuts the two strands. Okay. So yeah, this is the overall step of your elongation, and then comes your termination. So what happens is in prokaryotes, as you know, it is going to be a circular DNA. So they begin here and they both run in opposite directions. As I have shown here, I told you to consider only one half and forget about the other. So now imagine they both are running in opposite directions and they come back and meet each other at this point. Okay, so when they finally come and meet at this point, what happens is they split into, the next step of this figure would be to show two double-stranded circular DNAs. So this process does not need a particular termination, right? It just completes and it peels off as two different DNA strands, whereas our guy comes into picture, which is your eukaryotic DNAs. So as I've told you, I'm going to tell you, cover the eukaryotic DNA replication at the end. I'm just going to tell you the difference or an improvisation of how your eukaryotes differ from your prokaryotes, right? So prokaryotes, it initiates, the replication fork is opens up, and then your helicase runs, your topoisomerase keeps unwinding it, your DNA polymerase puts the pieces into place, and then finally two strands come into picture. One is going to go in the direction, the other strand is going to go opposite direction. So the opposite one forms gaps and then they adjust it together. Finally, they are forming into one piece. So this is there your prokaryotic DNA replication ends. And one more enzyme I have to mention, one more enzyme I have to mention here at the end of your lagging strand. Your lagging strand's gaps are filled by an enzyme called DNA ligase. So your ligation is a process in which you stick ends together or you tie up ends. Okay, so that is called ligation. Ligate. To ligate means to tie up or to stick. And so the enzyme DNA ligase, what it does is it sticks the portions of these fragments. The Okazaki fragments are finally stuck to each other by another final run of enzyme, which is called your DNA ligase. Okay, so there ends your prokaryotic DNA replication. And there's not much to cover in your eukaryotes. I'm just going to tell you the difference between your eukaryotic replication from your prokaryotes. Okay. Okay. So what happens here is, people are still actively researching to find out how exactly the eukaryotic DNA splits, because it's really complicated. It's really, really complicated. Then eukaryotic replication is characterized by many replication origins. That is the one primary difference that they have found out. The prokaryotic DNA has one origin of replication, whereas your eukaryotes have multiple origins of replication. As you can see, they have thousands, right? Then you have DNA polymerases. In your prokaryotes, the DNA polymerases used are DNA polymerase 1, 2, and 3. whereas your 3 is the one which actually performs the function. Your 1 and 2 are for repair mechanisms and for exonuclease activity. Okay, so repair mechanism is when finally it runs, it finds out any gaps in between, and if there is anything with a mismatch, what happens is your DNA polymerase 1 goes and DNA polymerase 2 goes and fixes it. So that is called the DNA repair mechanism. And in your DNA polymerase 1 is for you to perform exonuclease activity, which is used to remove the RNA polymerase at the end. So you know the RNA primers which start the reaction, they have to be finally taken off, right? They are only to start. It is like the person who actually does the wedding for us. Like in Hindu practices, you have your person who comes and offers the puja, or in other practices you have the person who actually commemorates the wedding, right? That person's role is only to begin. So, and he goes out of the picture, right? So the RNA primers have to be removed at the end. So that is done by your DNA polymerase 1. Okay, so it has exonuclease activity. But the difference between prokaryotes and eukaryotes is, here in eukaryotic replication, there is only 2 classes of polymerases involved, which are your polymerase sigma and polymerase epsilon. Okay, instead of 1, 2, and 3. That is the one difference. And then another difference, as you know, bacterial chromosomes are circular and eukaryotic chromosomes are linear. Everybody knows this. And why did I put this in a difference is because that is the major difference which causes the bigger trouble in the next step, which is in linear DNA replication, what happens is there is always a small gap left at the very end of the chromosome. And why is that gap left? It is because of the lack of a OH group for replacement nucleotides. And why is that gap or lack of 3-OH happens is because there is no RNA polymerase attached, right? So your RNA primer has to be removed from that place, and that leaves a gap. So as you can see here, this is a complete DNA, and then once it is split, it forms 2 copies here and here. So what happens is there will be a primer here and there will be a primer here. And after your process is done, the primers are removed, leaving behind a gap. This gap has to be filled in because they are linear. They are not circular. If they were circular, this end and that end will join together, so they are done. But here they are not circular. They are 2 different tracks, like in a single way, one-way road. So once they go, they go. So what happens is they have a gap in here. So how do you fill these gaps? Is when another person comes into picture, who is your telomeres. Telomeres are present only in eukaryotic DNAs, and they are overhanging DNAs which have repeated sequence of your A's and T's. Okay, so here you have an end. Okay, so here after after the primer is removed, the DNA strand doesn't know how to proceed further. So what happens is another enzyme comes into picture, which is called your telomerase, which comes and shows the direction, or like a guiding light for this sequence to grow. I mean, in what direction to grow, or what has to be there. So what happens is once it comes and attaches here, there is another gap left. So it is easier for this cycle to grow because it's from 5 to 3, as you can see. This will never grow. The 3 to 5 can never grow, right? So it needs some more space for the primer to come and attach. So the telomere will attach, and then this gap will be filled by your TTG, and then the telomerase will keep moving, giving you space for the new primer to come and attach here. So the new primer comes and attaches. What happens is this will grow. The Okazaki fragments will grow, and then the ligase will come and join them. The process is called ligation. And once they both attain equal length, or like an overhanging length, this is an unwanted sequence. It is present only for this purpose. So when you know that it is an extra, there is no harm in just leaving them alone. But this portion is an essential sequence which needs to be replicated, right? So what happens is if you have some extra, okay, you know that in the first step you have some extra. So you think that, okay, we have extra, so just leave it like that. But what happens is in every step of replication, 1 splits into 2, 2 into 4, 4 into 8. At the end of 8th step, what happens is this big extra, when it was there in the beginning, at the 8th step, it will become only this much, which stops you from going from 8 to 16. So that is why this telomere end has to be always, left extra, so the process can keep going on. Okay. So, yeah. As in a summary, as you can see, the DNA replication can be just summarized into these 10 steps. DNA unwinds and forms the origin, and then the helicase opens up the DNA and it forms a replication fork, and it's extended in both directions. And then the single-stranded binding proteins come and coat the DNA so that the rewind does not happen. And then your topoisomerase avoids supercoiling. The process is called supercoiling, which is your overwinding. So on one end to relieve the stress. And then your primase comes and produces primers. The primers are complementary to the DNA, which begins. And then the DNA polymerase comes and starts the reaction by binding nucleotides to the 3' end. So it grows from 5 to 3. And then the elongation of both lagging and leading strand happens. Finally, the RNA primers are removed and the gaps are filled with DNA by the DNA polymerase 1. And finally, the gaps in between the fragments, the Okazaki fragments from the lagging strand, they are sealed off by your DNA ligase. And there ends the replication of your DNA in prokaryotes. And in eukaryotes, the only difference that so far people have found out is regarding your linear polymerization, and it requires the ends to be taken care of, which is done by your telomerase and the telomeric ends. Clear? So there is a YouTube link that I have attached with you guys, so you can go and take a look at it later. And this one, I just saw it was a link which will actually show you an interactive session of how an actual the helicase and gyrase functions. Okay, these two links, maybe I'll post it in the chat box, or maybe I will send it to your professor so they can share it with you. Or you can just find it on your internet. Okay, yeah, that's about your DNA replication. And you guys have any questions so far? I know it's a Saturday morning and I've bored you enough for the past 1 hour. And good that I was able to finish it within 1 hour. I didn't take much of your time. Yeah. So students, if you have any doubts or any questions, not just regarding your DNA replication or anything that you wanted to ask, you can unmute yourselves and go ahead. And before that, I just have 2 more slides which they wanted me to show what we actually do in our labs. So I work on early detection of cancer metastasis. Metastasis is nothing but once we say that the person is cured of cancer, and after a few years, the person again develops a cancer, that is called cancer metastasis. And that metastasis is caused by very few population of cells, which are called as cancer stem cells. And we are working in our lab to find out or to detect those stem cells in a very early stage, so we can prevent the relapse of cancer. So we are using nanoprobes, which we synthesize using ultra-short laser pulses, and we use it to do that earlier detection. And also we have therapy and monitoring of cancer relapse, which another group works on. And also my recent work was on uptake of these nanoprobes by the cancer stem cells. So overall, people have found out only about cancer cell uptake, but what we have done was to show the cancer stem cell uptake of the nanoprobes, which can open a gateway to a new generation of cancer nanomedicine. So that's what I do for my doctoral thesis. And otherwise, recently I have been enrolled in one of the projects for this firm called Bombardier, which is your like a flight building aerospace company which does aircraft cabins. So we are trying to test their coating materials if they are working against COVID-19 or not. So here what we do is we just perform the RNA isolation of the viruses, and then we amplify that RNA into a cDNA, and then we count the number of viral copies. And after treating this virus with the coating materials, we were able to show a substantial reduction of viral coatings or like viral copy numbers, which is a very good result. So we can all, you know, we have that safety concerns when we are flying in the aircraft, especially amidst a pandemic. So that's what we are working on. So the RNA isolation and amplification is more of what I was just talking to you guys right now. It is more a resemblance of your DNA replication, just that DNA is a double strand, whereas RNA is a single strand, and we use whatever happens inside a cell or inside a nucleus, we try to mimic, or we try to do that outside the cell in the tubes by giving that conditions and giving all the enzymes. So in a tube, you add all your what is that, polymerase, and then your DNTPs, and then your exonucleases, and then you load them into different temperatures, and then you get a lot of DNA of the same thing that you gave. So it is literally the replication that you do. So replication is the very, very important and a basic step for your molecular biology. Only then you will be able to understand transcription and translation, and then your further PCR and other techniques, which you will be learning, I suppose, in your higher grades of your science degrees that you're pursuing. Okay. And yeah, thank you everyone, and thank you all the frontline workers who are working hard to fight the pandemic. And thank you all for listening to me on a Saturday morning again. Okay. Yeah. So it is 12:32 a.m. here, and I'm done. So if you guys have any questions, you can, yeah, go ahead with the questions. Thank you. Thank you very much for the opportunity once again. Sindhu was my very good friend, and we both met at King Institute of Preventive Medicine, where we were doing some project on, yeah, the influenza viruses. So I guess me and Sindhu are very much connected with viruses, like in the pandemic, like now in coronavirus. And at that time, swine flu was a hit. So yeah, thank you so much. Thank you again for the opportunity, the HOD and all the faculty members and the students. I appreciate all the efforts that you guys are taking amidst the pandemic to keep things going. Okay, thank you.
Thank you, Ashok. And really, it was a very nice presentation, very basic presentation, which our students will understand. Every time when we fix a guest resource person, we expect this kind of basic information, which the students will understand. But many a times we don't get such basic information, and it goes above the head of the students' level. But today it was really very nice. You touched the basic, and I think our students would have really listened, and it was to their reach to say the truth. And this 1 hour was completely fruitful, and I'm really very happy, because we never get such a resource person who touched the basic to the students level. So thank you, Ashok, sparing your time, that too in the night time. And elaborating them very in a detailed way, and making them aware of the things happening in replication. And we are also very happy that you are working with COVID, and we pray that you all are safe. Thank you, Ashok. Thank you.
Thank you. Thank you so much, ma'am. Yeah. So yeah,
students, if you have any queries, you can definitely ask him.
yes,
Ashok, there is a message in the post chat box also.
Yeah, I just saw it. Thank you,
okay.
Ms. Raghavi. Yeah, thank you. Yeah, if you guys have any questions, you can just post it in the chat box also if you are not comfortable in unmuting yourselves and talking. Or if there is no questions, then I take that as my presentation was very comprehensive and it was very explanatory that you guys didn't have any questions about it.
Students come forward. Definitely, the explanation will also be very simplified in a simplified manner. I'm very sure about that. So please do come out with your doubts. In case if you don't have any doubts, you can come up with the clarification of how he went to Canada, or what kind of exams you have to clear, or how you have to prepare for going abroad. So you can also come up with those questions also.
Yes, it doesn't have to be about DNA replication. It can be about anything that we have spoken in the whole 1 hour. So it can be about how to go about your higher studies, or it can be about how to do after your studies, or it could be anything within my expertise, so that I would be very happy to help you guys. Okay. And Nivedita, thank you so much.
So good morning, sir.
Yeah, good morning, ma'am.
Sir, it was a very nice way that you taught us, and the concepts were very clear. And thank you so much, sir.
Okay, thank you.
So, can you just please tell me that what PG degree can we choose after studying UG biotechnology? Only pure we have to take only pure biotech, or microbiology likewise?
So one thing I would say is people, actually you guys are a little bit in a better place than people who are doing their specific degrees like BSc microbiology or BSc biochemistry, though they have already narrowed down their path in their undergrad itself. But you guys are not like that. You have a very broad undergraduate where you will have a lot of options to go about. You guys can take your biochemistry as a major, or like molecular biology, genomics, genetics, informatics. There is like a wide range which you guys can go about, right? But those guys, it is a little bit difficult for them to choose any topic, but it is always good for you guys to take a specific thing which you like. So just give it some time. Do not focus on anything. I don't know which year you guys are in. If you guys are in your second year or in the first year, just learn every subject in which your teachers are teaching, and then try to develop interest on a few topics, right? So that you will know what to go from here. Don't stick to something which you haven't seen yet, right? So you never know. You might be suddenly interested in something else. No human mind is attached to something more than 6 months, or you don't have the same opinion about something or some person for more than 6 months, right? It's going to keep changing as you see new things. So what happens is go and have an open mind about all the courses that you're going to study, like the basics, like molecular biology, microbiology, cell biology, and your genetics and informatics, and all these courses. And then you can go about. And if you are interested in everything, or if you still haven't made a decision about what to go about, I would always recommend you to go for a master's also in a biotechnology, so that there when you do a thesis, or there when you do a final year project, you will be interested in one topic, and from there you can go about and make yourself narrow. So narrowing has to happen only when you go at the top, and in the base it always has to be a very broad spectrum. Okay.
Thank you, sir. Thank you so much, sir.
Thank you. And Meena Biga, the question is how, when, and where do cancer cells metastasize? So yeah, it depends. So for example, your pancreatic cancers can go and metastasize in your liver, and your cervical cancer can go and metastasize in your blood, or your stomach cancer, like your colorectal cancers, can go and metastasize in your kidneys. You never know, right? So it is all about which is the more susceptible place, right? So your stem cells look for a weak spot in your body, and once it identifies the weakest spot, or like it knows the ambient condition in a body where your stem cell can grow and attach, it goes and attach. It doesn't have a specific location or a specific organ in the body where it can go and metastasize. Yeah. Why did I choose Canada for my career? That's where I got, so I didn't have a lot of options. So Canada was my last option. I had applied for many other places, but then finally I got in 2 places. One was in Hong Kong and the other was in Canada. So I thought Canada is a good place. It's immigrant friendly country here. So people do welcome young minds from other countries. So they want people, because population here in Canada is very less, and they want working hands and thinking minds, right? So yeah, that's when you know your future, you have a better future in a country. So that's why I chose Canada for my career. Mithila Shrikant. Yeah.
Sir, good morning, sir.
Good morning.
Sir, now you said I applied, I got Canada, like that you say, sir. How can we apply for that abroad opportunity, sir?
Okay, so if you guys are looking for your masters, you can directly go ahead and look into the websites of the universities, and you can keep applying. So it has a lot of application links in all the universities. They have an option called Apply Now, and always go and look only for international applicants, and then it says how to apply and what to do. But for your master's program, the essential exam that you have to do is your English language exam, which is your IELTS. So you have to get trained, or you just have to practice it and take some online tests and then apply for the exam. And with that score, and also with your undergraduate score, you can always apply for the master's program. Or there are lots of agencies back in our country where people send people to Canada and countries like this. You can also go and consult them as to see for your score or for your area of expertise what universities would be of interest. Okay, you have to look for universities and then department, and then you have to specifically apply. It is not you will apply for a university, you will apply only for a particular course. If you are applying for a Masters of Science in biochemistry or Masters of Science in biomedical technology, you can apply only for that program. So if you have more than 2 programs in one university, you have to apply for them separately. Okay.
Thank you, sir.
Yeah, that's about it. And what is the scope of biotechnology in Canada? Biotechnology has had scope, will have scope, and will always have scope, because that's what people told me when I was studying, and that's what I tell for people as well. And then are there any specific exams when we apply for colleges in abroad? For US, it's going to be TOEFL, the English language course, and GRE. And for Canada, it's going to be, and Canada and many other countries, it's going to be IELTS. And I suppose US is also accepting IELTS. So it is better to go for IELTS for language and GRE. GRE is needed in all the European countries as well. And for biotechnology, if you guys are applying, apply for European countries. They are really good, and they have 1-year programs also, and people get admissions very easily. Recently, after this policy changes in Europe, they are admitting a lot of students, and apply for countries like Denmark, Sweden, and Norway. All these places are really good for biology and life sciences. They are really doing wonders. And you can always also apply for New Zealand and Netherlands and all the countries. Okay. And if you get a better opportunity in some other place, what will you do? Which place will you suggest for us to seek your career? This is more like a subjective question. So it completely depends on you. So you might as well stick to India. India has a lot of opportunities for biotechnology, like you have for Indian Academy of Sciences, Indian Institute of Sciences, and IITs. They are all doing wonders in biology, which abroad countries can never even compete off. Okay, so you never know. Country does not matter. It is about what you do and what you want to do. Okay, it is what it matters. And for me, to be honest, if I get a better option in India, I would definitely move back to India. Or if it is going to be China or Hong Kong or New Zealand, Netherlands, I don't know. Wherever I feel like my research is going to flourish, or I can do my individual research and nobody can sit on top of my head and instruct me what to do, I will go to that country. Thank you for the question, Mithila. And is it good for us to choose abroad for higher studies or for work? Yeah, so I think that is the same answer that I have for that question as well. And I guess I answered Sharina's question as well. And yeah. Okay, that was a lot of questions. Thank you. So if you have, yeah. I never expected questions. I thought you guys would be, okay, thank you for your explanation. When will the world get free from Corona? That is really a trivial question. We never know. We thought that we got the vaccine and we were literally going to be out of the pandemic. That is when we had our UK guys spreading the beautiful new version of it, 2.0. So we never know. We never know. What's the progress of coronavirus research that I am carrying out? Yes, the coatings that we have from the company, they are very well effective against the coronaviruses. We were able to substantially reduce the viral load in about 85% so far, but we are trying to move it up and ramp it up to 99% so that it's an absolute safety issue. Thank you, Sharina and Nirmaleshwari. Yeah. Thank you, guys. Sindhu?
Yes, Ashok.
Yeah. So I guess students are run out of questions.
Fine,
Yeah.
Ashok, fine. Yes. Ma'am, shall we go for the next thing, ma'am?
Yes, ma, we
Yes.
proceed.
Yes, okay. So, before
Sindhu,
going into
one
the...
minute.
Okay.
I just want to ask Ashok. Ashok, what is the current situation of Corona in Canada? Because we get a lot of informations like telling that the second wave is on and everything is going on, everything is locked down and other things. So, what is the true status?
Actually, what happened now was we are in a emergency lockdown, which is another third stage of lockdown which we have gone into. Last summer, we were actually going back to normalcy, but what happened was suddenly there was a hit and the ICUs were not able to handle the number of patients here in the country because the number of ICU beds were very less, whereas the number of patients every day were increasing. So what happened was the Toronto government had imposed emergency lockdown. So it is more like a curfew. People cannot go out of the houses from morning. I mean, only they can go out from a particular time. They can go out for other things, and all the stores are shut down. Only groceries and essential stuffs are working. And people like us who go for essential services and research, we can go out of the houses. So that is the state now, and it's really bad. Yeah.
Thank you, Ashok.
Thank you, ma'am.
Okay. So, Sindhu will proceed, ma.
Yes, ma'am. Okay. So before going into the formal vote of thanks, I just want to say a few words about Ashok. So we just met in King's Institute in Guindy, Chennai. So we started with a project, PG project. We started working in virology. So as Ashok said, there is a connection between our team. So it is not only me and Ashok. We have a crew of friends. We worked in a virology department in King's Institute. So again, it is coming to COVID, and we have a great contact, and we are to the next level. So thank you, Ashok. I hope you also got back the memories. We used to work in late evenings for our project works, and I think you remember all those places. So So thank you again, Ashok, for bringing back the old memories also.
Thank
So
you.
I
Thank you.
hope,
It was a pleasure.
I think most of our friends would be very happy that because you being a resource person, thank you, Ashok. So I just want to give a formal vote of thanks.
Okay.
Is I'm very much delighted to propose a vote of thanks at this informative lecture, Replication Pattern in Prokaryotes and Eukaryotes, organized by Biotechnology Department. And this program was sponsored by DBT Star College Scheme, Department of Biotechnology, Government of India. First of all, I just want to thank our correspondent, Thiru K. Palanisamy Avargal. He's I just want to say that he's a person who always motivates us in each and every activities which we do. And I just also want to thank our principal, Dr. N. Raman sir, who's always a pillar of support and guides us with all his experience. Next, I just want to extend my heartfelt thanks to our valuable resource person, Mr. Ashok Dhinakaran, doctoral research candidate in St. Michael's Hospital and Ryerson University, Toronto, Canada. For his immediate consent, because I just asked him a few days before, and he gave his immediate consent to give this lecture, even though it is late night and he has lots of works in his lab. So I thank you, Ashok, for giving us this opportunity. So your talk. This talk and your session seems that it is not a late night and it was like a fresh morning. I'm very much glad for bringing back all those things and giving a great lecture on this particular topic. Thank you so much, Ashok. I'm also delighted to thank our department HOD ma'am, Dr. C. Deepa ma'am, for friendly and positive approach in all the activities. And the next thing, I just also want to thank our DBT program coordinator, Dr. A.K. Vidya ma'am. She always leads us in a successful way. I just also want to thank other HODs of other departments. And the next thing, I just want to extend my heartfelt thanks to other fellow faculty members for their cordial support to complete this program in a successful manner. And last but not least, I just also want to thank my dear students for your polite listening. And I'm sure that this lecture would have enlightened you with the maximum information. So thank you so much, students. And once again, I thank each and everyone for bringing this program a successful one. Thank you so much, students. Thank you, everyone. Thank you all. Thank you.
Thank
So
you.
stay safe. For this is for the students and for my dear friend Ashok. So I think you have shared the link for the benefit of the students. So thank you, Ashok. Thank you so much.
Thank you, Sindhu. That was a very elaborative and thank you very much. And that was a pleasure to be with the students. And if you guys have any questions, you can always reach out to me at my email. I'll just post them here for your reference. And yeah, thank you guys.
Shall we end the meeting, ma'am? Okay. So thank you, Ashok. Thank you for joining us. So stay safe. We'll meet in another occasion. Thank you. Students, thank you. You can all leave.